introduce you to the students briefly, then you will start your lecture. Sudarshan?
The recording has started.
Okay. Good afternoon to everyone. I welcome all of you to the 36th lecture in the lecture series in Nonlinear Dynamics, conducted by the Department of Nonlinear Dynamics, Bharathidasan University, with the support from RUSA 2.0. I am happy to introduce today's speaker, Professor Sumana Datta from IIT Guwahati. In view of students assembled here, I would like to read out Professor Datta's CV very briefly. Professor Sumana Datta did her MSc in IIT Guwahati and completed PhD degree at Indian Association for the Cultivation of Sciences, Kolkata. She had carried out postdoctoral studies in Florida University, United States, about 2 years, and started her academic career as an assistant professor at IIT Guwahati in the year 2011. And right now, she is serving as a full professor there. Professor Datta works on various aspects on reaction diffusion systems, including spiral and scroll wave dynamics, and the system's response to the external stimuli and so on. Interestingly, Professor Datta also performing experiments on chemical reactions that display nonlinear phenomena. I came across Professor Sumana Datta's talk in a conference held at Jaipur, Rajasthan. It was a wonderful talk. After the talk, I noticed that several participants appreciated the talk delivered by her. So when I started this lecture series, I decided to request her to deliver a talk for the benefit of our students, which she immediately agreed. Since I don't want to take much of her time, now I invite Professor Datta to deliver her lecture. Over to you, madam.
Thank you so much, Professor Senthilvelan. It is indeed a great honor to be associated with Bharathidasan University and Nonlinear School. It's something which is extraordinary in India, and it's also, I'm honored to have the presence of Professor Lakshmanan and Professor Shamal Dana also. Thank you so much. I will try to share with especially our students here today the kind of work we do. And before that, I would like to take them through a journey of chemical systems which show nonlinearity, and why we study them and how we study them. So I will just start sharing. I hope this is visible.
Yes, ma'am. Yes.
So I entitled the talk Spiral and Scroll Waves, Exploring the Nonlinear Behavior of Chemical Oscillators. So first, as I generally show some general pictures to show patterns in nature, I was told by Professor Senthilvelan that Professor Devshankar Rai, who was my PhD supervisor, has talked you through pattern formation in chemical reaction diffusion systems. So a little bit of that will overlap with what I'll show. But these are some pictures that I have taken of different patterns that we see. Sometimes we observe them in nature, sometimes we overlook them. But if we look nicely, if we really observe them, we'll be awed by the kind of patterns that we see. So patterns in clouds, this is a kind of a stalactite, stalagmite pattern that is formed in caves. A lot of these caves in Meghalaya are there. There are several patterns in vegetation, patterns on the wings of insects or the skins of large animals. Now, how these patterns are formed generally are studied by mathematical biologists, and the process has been coined by Turing as morphogenesis. So it is changing, and with time, how the patterns change. So this is the picture of a fish at 10, 12, and 13 months age. And below the lower panel is actually a numerical simulation of some differential equations which show similar kind of pattern, and how this pattern, which you see a fork-like pattern, it slowly disappears. Actually, it moves from the left to the right, and finally you get only parallel lines. So these kind of systems are studied by biologists as well as mathematicians or physicists, and these patterns are called spatiotemporal patterns. So this is Turing, and I am sure you all know about Turing. So he was the first person to give us mathematical equations for explaining all these kind of systems which has similar patterns. So systems that go from hydrodynamics, clouds, to patterns on fish and animals. And one interesting outcome of this was the Turing-Murray theory, which says that an animal with a spotted body can have a striped tail, but the opposite is not possible. That is, an animal with a striped body cannot have a spotted tail. It is true in nature, and the reason is that this diffusion and the boundary condition. So these two things are important in spatiotemporal patterns. Now, when we talk about all these patterns, of course, there are mathematical models that you know, some of them, some we will talk about. But then it would be nice to actually have mathematical laboratory models to help to understand that the theory that mathematicians put forward, whether that is true. And here chemical systems come in. So first, I will give you some glimpses of different chemical systems, not necessarily one showing reaction diffusion, but systems of chemistry to understand different natural phenomena. So this is one example of chemical garden. So it is a silicate kind of precipitation tube, and they are used to understand the formation of geological patterns or geological formations like soda straw and then mud volcano, etc. Not only does the chemist stop there, they also use this kind of understanding in using different kind of in use and they use this physics that they get in generating different kind of devices. For example, this over here is kind of a fluidic device that they have used by optical control of this kind of microtubes. And on the right corner here is basically, this is also experimentally obtained micro or nano structures that are aesthetically pleasing and also have very thin tubes which can be used for other purposes. Chemistry is also helpful in understanding larger geological formations like these laser gang rings that are found in rocks. So on the right hand side is a picture of laser gang rings formed in a test tube. Now, depending on the size of the test tube and other temperatures and other things, we can actually form this kind of rings in the laboratory and try to understand how these formations have occurred in the world across millions of years, right? And then this is a topic which you are very well acquainted with, because I'm sure a lot of the groups at Bharatidasan also does synchronization. So if we think about synchronization in nature, the synchronization of fireflies, chirping of cricket, these are things that we generally say. In chemistry, there are examples in the laboratory, but before that, I'll just give you one simple example. If you think of a soap, which we use in our day-to-day lives, so that has a long polar tail with a small non-polar, basically a long non-polar chain with a small polar top, this one at the end. So what happens when you put soap molecules in water? They form kind of a shell like this. This is called a micelle, and inside is your non-polar, apolar part, and outside is your polar region. So it's soluble in water, and what happens is inside it holds the oil and grease which is otherwise insoluble in water. So this is how things are being used, and this is synchronization in chemical oscillators. So these are experiments done by Professor Puneet Paramananda at IIT Bombay, where they use synchronization in mercury beating heart system, showing different kinds of patterns, and also chimera-like dynamics in camphor rotors, which shows clustering and chimera. So self-organization, when we talk about, that does not stop with something which is visible to the naked eye. We can also go small and look at biology, and the DNA that we are all formed of, or that which forms the basis of all biological individuals, right? So those DNA are also self-organized. They form these helices, and that identifies an individual. How do chemists use it? So they take up this knowledge and the knowledge of fractal geometries and make these kind of big molecules, which acts as a cage to carry medicine inside the human body. So these are dendrimers and they are used for drug delivery. So this has been inspired from fractal geometry. Now coming to the kind of systems we are more interested in are chemical systems that show pattern. First of all, this is an activator inhibitor system showing Turing pattern, another chemical system, and finally another chemical system where Turing pattern is driven by light and the pattern changes from with the intensity of light. Now, these kind of spatiotemporal patterns in chemical systems are found in a few interesting examples. So one is the thiourea iodate sulfide system and another one is the CIMA or the chloride iodide malonic acid reaction. In both of these, you can see with the same concentration or the same species of chemicals that are added together, they can see spots and stripes, just like we saw in the in the animal's skin, right? The skin pattern. And we should remember that whether they be cells or DNA or whether they be patterns on the skin, finally biology is chemistry. Okay, so that's why understanding them with a model in the laboratory becomes ever more interesting. So coming to chemical waves, I would like to talk to you about the Belousov-Zabotinski reaction. So Boris Belousov was the person who was trying to find actually a different reaction. He was mixing a few chemicals to find Krebs cycle. If you have studied that, that's an organic reaction. He was trying to make an analog in the laboratory. So that was 1953. And what he found was the solution changed color between yellow and white and colorless. When he tried to publish it, nobody believed him. But then in 1961, Zabotinski came ahead and he changed the chemicals slightly. What he did now was change this cerium sulfate to heroin. And what we see is this. When you mix all the chemicals and stir them, maybe with a glass rod, maybe with a magnet, the color changes from orange to blue. So now scientists couldn't really say that there was some impurity and maybe this yellow color is something that just happened. But then people did not accept it still. What was the reason? The reason being all the thermodynamics that we study in our BSc, MSc, all of that is close to equilibrium. So when different chemicals react and they reach equilibrium, why should they again come back? That was the question. So people thought this went against the second law of thermodynamics. So Professor Belousov could not publish it and he died an unhappy man. But the concept of this equilibrium was kind of challenged in a 1961 book, which was entitled Introduction to Thermodynamics of Irreversible Processes. And the concept of dissipative systems or systems which cannot dissipate heat or systems far from thermodynamic equilibrium was brought into the picture. So what is actually? So if we consider this is the potential energy diagram of any reaction that is happening, okay? And this ball is the state of the reaction at a particular time. So as it goes from the top down till the time it reaches the global minima, it can actually stop at some points. So these stoppages are not the global equilibrium. This is indeed the global equilibrium, but it can have some steady states other than that too. So this was a concept unknown to the people at that time. The person who dared to look at something out of the ordinary was Ilya Prigogine, and he won the Nobel Prize in Chemistry in 1977 for bringing this concept of systems far from thermodynamic equilibrium. So if you look at the Belousov-Zabotinski reaction, I'm not going to go into the kinetics because it might scare you. Basically, it's an autocatalysis. So you have heard about catalysts that mainly speed up the rate of a reaction. So autocatalysis is something like when you learn about nuclear fission. You have neutrons in 3 neutrons forming, right? And then they go and hit the next atom and again they form. So things like that. You see this molecule marked in red two molecules are formed, and that again takes part in the reaction. So the product, one of the products are again taking the chain forward. So that happens. When that happens, then interesting things start to happen. And then the linear dynamics becomes very non-linear. And here it is the ferroin, the catalyst, that changes the color from red to blue and back. So now I can show you something which you may be more familiar with. When we write the mathematical model of the reaction and we take these as simply variables, forget that they are concentrations. Now think of them as simply variables, and you solve these variables. Okay, you look at how they change with time. The x, y, and z, you see that they are oscillating. So they are periodic, they're going up and down. So you can see that they have a time period, they have an amplitude and everything like the regular oscillators do. So before I move into the physics part, let me show you how the busy reaction is carried out. So this is
On the experiment.
a tiny video. So what we do, we take sodium bromate and pour water. So we make a solution of sodium bromate. After that, we keep it aside. Then we take malonic acid and we make a solution of malonic acid, or in this case, malonic acid and sulfuric acid together form a solution. And that's the second solution. Sodium bromide you may or may not add. And then you take a ferroin solution which can be bought from any of the chemical companies. Very simple things which is available in the undergraduate laboratory. Then you take a beaker, add the two solutions 1:1 ratio, that is sodium bromate and malonic acid. And here the sulfuric acid was already there. And start stirring it. If you don't have a magnetic stirrer, you can stir it with a glass rod. And then you add the ferroin solution, which is already red in color. Now, when we add that and stir it, you can see after a while that the color starts turning blue, okay, and back to orange again. And then it starts oscillating back and forth. So here it is shown that they're turning the heater on. So increasing the temperature actually facilitates the reaction and makes the rate faster. So you have a faster oscillation period. And you can see how beautifully the color goes from orange to blue and back. So you can see for yourself that that is oscillating. This is another reaction with hydrogen peroxide, iodate, and sulfuric acid. Everything has been added and you are stirring it. And you see the color goes from blue to yellowish, back to blue, dark blue, and back again. So these are undoubtedly oscillating. This particular reaction is also known as the iodine clock reaction because the time period is so fixed that you can actually keep time with this kind of reaction. And of course, you can change the time period by changing the different concentrations and stuff. So now what happens if we take this reaction, which is oscillating, and put it in a petri dish? Okay, so now, till now we were stirring it. So there was a homogeneous mixture. Now when we pour it in a petri dish, let us see what happens. So you can see the spontaneous formation of waveforms, right? So you can see that they are circular waves that are emanating and they are kind of going out in all directions. So these are called target patterns. And this is a beautiful phenomena that can be observed in the BZ reaction. So you can call them chemical waves. They are like waves or ripples in the water, but they are different from it because ripples generally die off. But here they are living on theoretically forever. In this case, not really so in a closed reaction. So what are these chemical waves actually? So we know that there is some reaction going on. Some reactant species are or intermediates are oscillating. We can see that also. We have seen with equations. But why these patterns? So these patterns form because of the interplay between reaction and diffusion. So what is diffusion? Suppose I have a plate of water and I drop a drop of ink in it, and then the ink is going to spread. So that is diffusion, right? So when reaction and diffusion interplay, then you get this kind of beautiful patterns. So writing in terms of diffusion equations, dc dt, where c is the concentration of any species, is equal to f of c. So these are all the kinetic terms. You need not remember what they are, but they are the kinetic part. And then there is a flux part. Okay. So the rate of change of the variable is equal to the kinetic terms, which is the reaction, and the diffusive terms, which goes into the flux. So what is the physical significance? The reaction is trying to take the reactants to the products, and the diffusion is actually mixing the two. So this is the competition that is happening, and that is giving rise to these beautiful patterns. Now, if we have only translational diffusion, that is only the particular species is diffusing in space, then we know by Fick's first law that the flux is given by minus d gradient of c, where d is the diffusion coefficient. So in 2 dimensions, we can simply write the second term becomes dc del 2c del x square plus del 2c del y square. So this is my differential equation, and using a proper reaction mechanism, maybe the Belousov-Zabotinski reaction or any other reaction, if we put all the details into FC, we can solve it using Euler or Runge-Kutta method and get what is going to happen. We can predict. So now let us see. We have different kinds of waves are possible, right? We can have straight waves, we can have curved waves, etc. So what is the effect of curvature on wave propagation? That is important for the spiral and scroll that we're going to soon talk about. So if we have a wave, it has a head and it has a tail. So an activation front. So if it's a straight wave, if the velocity is v, a concave wave has a larger velocity and a convex wave has a smaller velocity. As physicists, you will know that. Now, when I have a plane wave going, it will move with the same velocity all through because the curvature is same. If I have a circular wave, just like the waves we just saw, then too the velocity will be same and we will get concentric kind of circles, right? It's going to spread nicely out. But what if we have something which looks like a spiral like this? Then you can see the head is the red region or the activation front, and the tail is the blue region. And there is a point where the two meet. So that is the phase singularity. That is also called the tip of the spiral. And you can see if these arrows show the velocity of this wave, then the velocity is different at different regions of this wave. And at the tip, it is 0. And this tip actually, that's why, moves around a geometrical entity which is known as the core of the wave, the spiral wave. So how can a spiral be formed? We saw our beautiful concentric rings. How can a spiral be formed? Suppose I have a plane wave. It could be a circular wave also. And by some reason, we break the wave or it gets broken. Then what happens is this tip, it starts moving slower. Okay, the velocity here is slower than the rest of the plane wave because it is convex. And once it is slow, then this part moves slower than the rest and it curls in to form a spiral wave. So this is how a spiral is formed due to wave break. Now, why do we study spirals at all? So let me come to that part. So you know that the heart has a lot of problems sometimes. We have fast heartbeats and a person can undergo cardiac arrest. It has been seen that these kind of spiral waves have some relationship to these problems in the heart, mainly called tachycardia or fast heartbeats. So the spiral wave, if you look at the velocity of the wave, is different. So if you draw the action potential in different spaces, you will see that it is different and that is faster than a regular beat. And that is why these kind of waves are formed. Let me explain to you. So this is how the heart looks like. And here it has 2 upper chambers called the atria and 2 lower chambers called the ventricles. So in the right atria, you have something called the sinoatrial node or the pacemaker cell. We all have natural pacemakers in our heart. So these actually give off these electrical signals which make the heart contract and expand, contract and expand. When it expands, blood goes into the heart. When it contracts, the blood is pumped out. That is how the heart actually takes in the used blood and puts back oxygen into it, oxygenated blood, and takes it out. So this is what the heart does. And in 1 minute, you know that the heart can actually pump 5 liters of blood. So almost all of our blood goes through the heart once. And this oxygenated blood is carried to the different organs of the body. And that is how different organs get oxygen. So if the heart is to malfunction, then our whole system is disrupted. And we do know, sadly, that cardiovascular diseases are a leading cause of deaths all over the world. Now, what could be the cause of heart diseases? There are several reasons. First, coronary heart disease due to blockage of the artery. A person can have a stroke, which is nothing but the brain does not get any blood anymore. There could be weak pumping of the heart. That can happen when a person grows old or due to some birth defects. And then cardiac arrhythmia. So this is the disruption. Arrhythm means the rhythm is broken. This is the disruption of the normal cardiac rhythm. And that is mainly of two kinds, tachycardia and bradycardia. Tachy means fast. And this tachycardia is one of the most leading cause of heart disease, especially in a otherwise healthy person. So what is actually happening? When the heart beats, you can think of the signal as propagating, an electrical signal, and this is the voltage signal which is propagating here. If you consider this red dot as a cell of the heart, then this is the action potential that will be felt by this cell, okay, of the cardiac tissue. Now, this action potential is related to the different sodium channel, calcium channel opening, etc. Not going into the details. But what we see, or what the doctor sees, is this kind of an ECG of the heart. So these two are actually well related. All these are numerical work done by the group of Fenton and Cherry, and their group you can actually find online, and it is awesome. It's mesmerizing the way that they have actually brought these things together. And you can see that the upper chamber and the lower chamber, they actually go off, they pump the blood one after the other. And this sequence brings about this PQRS T, this signal. And even if you have not seen an actual ECG paper, you might have looked at this kind of diagrams in a movie or a serial. We always see, right? This kind of a monitor goes and the signal is seen of a beating heart. So now what are the problems that these spirals can cause? So how did we know these spirals are causing? Again, this group called Virtual Heart, they actually did simulations, and other groups too. And they found that if there is a source of a signal other than our normal pacemaker cells, a source like a spiral, right? If that is there, then these kind of signals will be formed. So these signals, these pictures which I have here of signals are actually ECG pictures. So if there is a flutter in the atria, heartbeats will go up to 350. So if the heart beats very fast, what will happen? That the total amount of blood is not going to be pumped out. So that is the problem. This can move on to atrial fibrillation, where the beats per minute is 600. For a healthy person, the heartbeat could be 60 to 90, right? If you're an athlete, it is 60. Somebody like us, it will be 90, 95. But 600 is something abnormal. These are life-threatening in the long run if these patterns, or if this problem goes into the lower or the bigger part of the heart. If the ventricles have tachycardia, then things are much more serious. If there is a single spiral, or in this case scroll, I'm going to tell you what that is, then you have this kind of a QRS complex. And if there are several such spirals, then we have ventricular fibrillation, and the person might die within minutes, if not immediately administered some procedure. So this is one example of an ECG of a otherwise person whose heart was beating okay, right? In the first few lines, each line is a minute of heartbeat, and then suddenly something happened here in the 5th minute, and then things started going bad from there. Things became chaotic. There was no more the signal. There was total arrhythmia. And then finally you see, you know what these flat lines are. These are the person has undergone cardiac arrest, and that's basically the heart has stopped beating. So this is a video, I'll go quickly through it, which shows here actually it's a swine heart, and the scientists are injecting potassium. So its sodium potassium balance is very important for the heart, and they accelerate the beating of the heart. And when it accelerates, see, the rhythm is broken from the normal sinus rhythm, and then this is tachycardia, and the signals on the live ECG, they keep on changing, and then slowly the heart beats become very slow. You can understand that it's hardly pumping any blood, and then finally it decelerates further and stops beating ultimately after a while. Okay, and you can see that that is cardiac arrest. The heart has stopped beating and the lines are flat. So that is the procedure of ventricular fibrillation. Now, what do we do when a person is having this fibrillation? If you understand a person is having cardiac arrest, generally electric shock is administered to that person. So this process is called defibrillation. So electrodes are placed on the chest and on the side of the heart, and electric shock, high electric shock is given in the hope that it is going to remove all these unnecessary waves, and the pacemaker cells of the heart is going to start again. So CPR is also given following that. I'm not going to the video. You can find the video online. Now, the methods of defibrillation are strong electric shock. So this electric shock is going to cause damage to the heart for sure. It's going to burn muscles, and those burned tissues are going to be future sources of such fibrillation. So we need better methods of defibrillating. So for that, we have to understand what is the process that is happening. Now, it is possible, it has been possible over a few decades to actually look at cardiac tissue under the microscope. But as you have seen that it is not really possible, very easy to work with a live heart or even a cardiac tissue for that matter. And people like you or I who can sit in the laboratory, maybe do some experiments or run computer simulations, it is really difficult. So we need models to understand the physics of these waves and try to implement that to the real system. So first, going through one of the most famous mathematical models, I'll not go in details, but I just want to show you some cardiac models like the Luo-Rudy model for the heart, where we have the variable here is the voltage, okay, the gap, the membrane potential, and the ionic currents. Then the Fizeau-Nagumo model, which is widely used, again the voltage is the fast variable and the V-gate, which is the slow variable. And another model like the Alif-Panfilov model, where the membrane potential and the conductance is considered. So mainly you understand the action potential or the voltage thing here is considered, and all the studies of the heart are carried out. Now the heart is what we call an excitable medium. So what is that? So it is a system which can allow a signal to propagate through it without damping. Now if we think the ripples in the water, if I drop a stone, the ripples will form, but it is going to die or damp down. Now my sound waves, when I am speaking, it is going to go up to a distance, but then it is going to damp. So these are not excitable medium. But when you saw the waves in the Belousov-Zabotinski reaction, the beautiful waves that were going, they did not damp. Is that excitable? Maybe it is. Is the heart excitable? When the heart pacemaker cells give off a wave till the end of the heart, they go, they do not die down. Yes, it is indeed excitable. So one important property of excitable medium is that they have a refractory period. That means after one signal has passed, it is to come down to its rest state before another signal can pass. Think of a forest fire, okay? So suppose there is a forest fire. You know that if you can cut off, allow it not to propagate, right? So suppose the entire area has burned down. Now, if I want to send another signal, another fire, the forest has to grow. So here the refractory period is in tens of years, right? But then in the case of BZ, you see one wave is followed by another wave. It is not that long, but it is long. Now, these are the properties of the excitable medium. Some other excitable media, other than the heart and the BZ reaction or the chemical waves, are the propagation of electrical excitation in the nerve fiber. I'm sure you must have had speakers talk to you about signal transduction in the brain. Okay, so those are also, the brain is also, and the nervous system is an excitable media. Propagation waves during the aggregation of social amoeba, concentration waves in glycolysis, and calcium waves in oocytes. Let me show you some beautiful pictures. Okay, so all of these are not waves for sure. These are cell patterns. Yes, they are waves, we can call them. The spiral galaxy is something that we all know about, and this, it's interesting that this also likes to take this spiral structure. These are calcium waves in oocytes. Amoeba population. So this is not a single amoeba cell. There are several cells of amoeba, several population of amoeba that get, they like to arrange themselves in this way when the food is scarce. Okay, this is a spiral in a neuronal tissue, and this is a spiral in a ventricle cell culture. So this is a cardiac tissue, and this is a spiral in a Belousov-Zabotinski reaction, which can be formed by breaking these circular waves. This is another chemical example of a heterogeneous catalysis of carbon monoxide by oxygen. This is another very breakthrough work by Professor Gerard Ertl, which earned him the Nobel Prize. You can see spirals of carbon monoxide and oxygen. And this is not really a spiral, it is a helix rather. So this is also formed from the same lysogang pattern that we saw. You remember the rings, the concentric rings? If we change some things, maybe make the tube where the rings were made smaller, larger, some change, it is possible to make a helix like the DNA. So we saw different systems now. Are they all the same? No, of course not. As we said, they have dissimilarities. Mainly the conduction velocity in the heart is in meter per second. In the calcium waves in the cell, it is micrometer per second, and in the BZ waves, it is in between, it is in millimeters per second. So you see it's 10 to the power 3 order difference in these. Moreover, the heart is inhomogeneous, discontinuous, and anisotropic. Compared to that, the BZ system that we saw is just some chemicals mixed together in water. So it's very homogeneous and it's also continuous. Now the challenge lies in bringing about these changes. Can I make it inhomogeneous? Can I bring in the discontinuity? But over and above everything else, we see that they are excitable media, and this action potential kind of propagation of signal happens in all of these. And most important is that both of them or all of these actually sustains our spiral waves. So we use this basic system as a model for the heart. So this is our simple experimental setup. This is the BZ reaction which we do in a Petri dish. It is illuminated from below with the help of a lamp, and we monitor it from the top using a CCD camera. And we store our images onto a personal computer. So this is the video of a spiral wave. Just see how the waves propagate, kind of. So this is the tip of the spiral, and this red dot is something that we have given. It is not present in the actual experiment. We put it while making the movie to show that the spiral tip is actually moving around a small area. Okay. So this is the tip trajectory which we have made with MATLAB. We have used to trace the trajectory, and we found for our particular chemical recipe, the core size is 1.09 millimeter. So these are snapshots of the spirals. At first, there was only one wave. So when one spiral, once it touches each other for one cycle, the one wave goes out, and then the core remains there. But one after another, waves are given out. Now, if we take a small section of this across all the times, through all the snapshots, and plot them like this. So this line is this, okay, horizontally here. Then with time, if we stack them one over the other, this is called the time-space plot. And you can see the waves once going this way. So when this spiral moves, it gives off, this wave goes there and this wave goes here. So it's like a fishbone kind of a structure. And from here, you can beautifully calculate the time period and the wavelength of the wave. This is where the spiral tip resides. Now, with all our mathematical model, our experimental model, till date, what did we learn from these models? First, we have learned that if there is a heterogeneity, that is, in our busy model, we can think of a glass bead, a small rubber ring, or a tube or something. A heterogeneous means the wave cannot pass through it. So if such an obstacle is there in the path of the plane wave or the circular wave, it will break and the ends will be free. They will curl in and form a pair of spirals. So in the heart, that can easily happen if you have a clot, if you have blockages, or if you have dead tissue or burn tissue or scar tissue due to the defibrillation technique burns some tissues in the heart. So that can cause more such wave breaks. Even without a heterogeneity like that, it is still possible to get the formation of a spiral. So when the excitability becomes low, that means what is this excitability of the system is how easily it can allow the wave to pass through it. So if it is all equal excitability, the wave is not breaking out, right? But in this case, the low excitability makes the wavefront detach from whatever it's attached to, and that can curl in and form a spiral. Now, even without anything, I said that if a person gets very angry, a very healthy heart, still he or she can have tachycardia or can have fibrillation even. What is the reason? The reason is that they have a refractory period. Now, when one wave passes, before another wave can pass, it has to come down to the rest. The rest period is there, right? It has to come back to the original state. If that time is not obtained by the system, if another wave immediately comes in its wake, it does not have enough space to move or enough excitability. This system has still not gone back to the rest state. So what happens is this wave is no longer a continuous wave. It moves slowly, and this slow movement makes a spiral form. Now, if these spirals drift as they do in experiments as well as in the heart, they change the pattern of the ECG. This was one experiment of the BZ reaction where we show a spiral wave or a spiral tip actually moving away because of some waves that are clashing against. So these moving waves actually give rise, because of a Doppler effect, to some pattern which is actually called polymorphic tachycardia in medical terms. So in general, if I summarize, what spirals have got to do with the clinical condition of the heart, a stable spiral wave which is moving in one particular point will give rise to an ECG like this, and it is called monomorphic ventricular tachycardia. As soon as it starts moving, meandering, which we say, it gets more complicated, the ECG, and it moves towards fibrillation. When it becomes chaotically meandering, of course, the signal also gets chaotic. And finally, when one spiral breaks up into many, that chaotic signal that is formed is really of very low amplitude, and finally that can cause cardiac arrest. That is, the signal might completely die. That is ventricular fibrillation. Now, what will happen if a spiral is moving? That is dangerous, right? What will happen if we intentionally put some obstacles there? Theoretically, it was suggested that putting another obstacle there can actually arrest the movement of the spiral. It will get pinned or attached to such an anchor. So a polymorphic tachycardia, which is more dangerous, will now become a monomorphic tachycardia. And it was also proposed because the doctors treat the patient, right, with high blood pressure, with high hypertension, with very fast heart rates. Injection is given, right? So that is tetrodotoxin is one of the injections, TTX. And what it happens is it's actually a sodium channel blocker. Less sodium means less excitability, and the waves become slower. So you can see that the core size increases and the velocity of the waves become low. Okay? So this is something that is achieved or done by the doctors without understanding basically what is the physics that is happening. So this is physicists telling them what is actually happening. So we know that eating too much salt can give high blood pressure. So you can relate that part. Now, one thing we do need to remember is that the heart has a 3D structure. And even though we can see what is happening in a tissue of a heart or the top of the heart, we cannot really visualize what is happening inside. What is happening is there is not only a 2D spiral, there is its 3D counterpart, which is called a scroll. So a scroll is nothing like a scroll of paper, nothing but like a scroll of paper. So if I have several spirals and stack them one over the other, then all the tips join together to form the filament of the scroll. So a scroll can be straight or it can be ring. When the filament takes a circular shape, then we call it a scroll ring. So the heart actually has scroll waves, especially in the ventricular part, where it is really thick muscle. And by numerical simulation, one can see what is the shape of the filament and what is the surface pattern. So if I have a straight filament, it looks as if there is a spiral, a single spiral head here. If this is the filament and that is the 3D shape of the filament, this is how the surface wave looks like. If the filament is a U shape or C shape like that, it looks like a pair of spirals. And if it is a round shape, looking at the surface, you cannot say anything. So the requirement for mathematical models and laboratory models goes up several folds due to this 3D structure of our excitable systems. So how do we generate 3D scroll waves in the BZ system? We first take one layer of gel. Then we put a silver wire. So this silver wire acts as a catalyst, local catalyst. There's a wave, a circular wave that is formed. After this wave is initiated, we pour another layer and we see that the scroll has formed. So what is actually happening? If you look at a cross section from the side, the silver wire gives rise to a wave which is going to go in all directions, right? So this will be like a bowl, okay? A bowl. When the bowl has reached a particular dimension, we pour a second gel layer on the top. So now this bowl has all these top ends which are free. So these ends are going to move into the top layer. They will go up and come down. This is how they will form the scroll, which will look like this. So they will be like the top, the tire, right? So if we look from the top, then what we are going to see is something like this. A wave going in and going out, going in and going out. So they are like going in and out. The spiral is going like this. So these are the individual spirals and the scroll behaves like this. So if we look at snapshots, what you can see here is if I say that this white area in the middle, this one, this ring is where the scroll filament is, then you can see that the filament actually shrinks. So this does happen in case of a system like the BZ reaction, where we say that the filament has a positive filament tension. I'm telling you soon what it is. Now, if we take a straight line across this scroll and plot with time through all the snapshots that you have taken, right? Then you will see that this is where one end of the filament lies, and this is where this line cuts the filament. So if we join them with two straight lines, we can see that the scroll is become smaller. The filament, the circular filament is becoming smaller and smaller, and finally it disappears. So for a diameter of a scroll of 7 millimeter, the lifetime is 2 hours. So this is the curvature flow model of filament dynamics, where S is the curve that you're talking about, N is the normal vector, B the binormal vector, alpha the filament tension. So if alpha is positive, and K the local curvature. So if alpha is positive, the ring is going to shrink. If alpha is negative, it's going to expand. Beta, on the other hand, is how this will move in the binormal direction. So a closed filament will always eventually become a circle if these are the velocities of the different parts of the waves, and an open filament will eventually become a straight line. So now comes the question. Now that we have chemical waves, now that we have spiral waves and scroll waves, can we do anything to control these at all? We know that electric shock is used in the heart system. Let us see what our mathematics tells us. So it has two parts, right? This reaction diffusion system. It has the reaction part, which has all these concentration variables. So can we change the concentration? Can we gain a kinetic control over our system? Can we change the excitability of the system, like the doctors did giving the TTX injection? And the second part is our flux. Can we change the flux in any way? Can we use an additional external flux? Can we make the system heterogeneous to control the dynamics of these waves? So this is the example I was talking about. So let us look at one of the models of the BZ, the Originator model. So here you see is a two variable model. It's a simplified version. U and V are the two variables, the inhibitor, the activator and the inhibitor. So you see the ferro in the catalyst is the inhibitor. So these are the kinetic parts of the equation, and then you have the diffusion, only the translational diffusion in this case. Now what if we play with this epsilon? So from previous theoretical works, it was suggested that epsilon is K5, K3, concentration of organics by concentration of acid and bromine. Now, what is K5 and K6? They are just some rate constants. Their values are this. The organics are nothing but the concentration of the malonic acid and the bromomalonic acid, two species. And we have all this F, Q, etc. If we take the acceptable values, we take some value of dt, of the diffusion coefficient u and dv, and over a space of 300 by 300, if we go ahead and do a Runge-Kutta method of integrating these equations, then plotting u as a function of space at a particular time, you can see this spiral wave. Okay, so this spiral wave dynamics is exactly like what you see in the experiment. And along the spiral wave, if we take a line, again like we did for the experiment, and plot it, we get this kind of a time-space plot. This is the time period, this is the wavelength. Now, what if we change this epsilon? Okay, epsilon changes from 0.02, 0.03, 0.04. So we are increasing the epsilon value, and we are seeing that the spiral becomes slower, its wavelength increases, its time period also increases. Now, also the tip trajectory, which we saw only round at first, right? Now this tip trajectory gives us very beautiful patterns, floral patterns. And this is the spiral core. And the size of the core changes. If we take the height to width ratio, it is found with increase in epsilon, the height to width ratio decreases. Okay. Now what happens with the frequency? So frequency is nothing, we are saying is just the inverse of the time period. So if we plot the frequency with respect to epsilon, what we see that there is at first an exponential decay, which is shown here in the inset better, e to the power minus epsilon over something. And then something happens over here, and then it goes up and remains kind of stable. So these red triangles are the beginning of an area of transience. I'm just going to talk to you about what this transience is. These and these are stable oscillations. And for this, because this is transience, we are taking the average amplitude of a system undergoing random oscillations in between these two transient points. So this area before the transience is where we get spirals for our reaction diffusion system simulation. Now, if we take our experiments, you see the dots are of course a little bit random. If we take our experiments and do the same thing, exact same thing, we have seen that the time period, inverse of the time period or frequency also, it has an exponential decay. Now these are numerical results of the phase space and the time dynamics of the variables. So U versus V gives here the limit cycle, which you can see. And there is a kind of an envelope over here. So this is for epsilon 0.04, which is this area. And this is beyond 0.12. So exactly at 0.12, you see it has a chaotic kind of a behavior. So this is a lot of, so if you take just at the end, you see it kind of reaches as a limit cycle, And this is the random part, and beyond the transients is something like this. Okay? And beyond that region of beyond 0.2, we are getting this. So stable oscillations, very nice limit cycles. And even further, when we increase epsilon, we see that the system goes to a steady state. So these sorts of graphs you must be acquainted with. So we can also look at the amplitude as a function of epsilon and amplitude as a function of frequency. So in the absence of the diffusion, there we are not going to have any kind of waves or pattern. So it's just a homogeneous system. In the experiment, we can think of a homogeneous system where the variables are changing. In the simulation, we know how we can plot V as a function of U, the phase space, and here we see the limit cycles and the supercritical half bifurcation at 0. So beyond 0.30. So if we draw it in the nature of a bifurcation diagram, here we see that at first we have stable oscillations without diffusion, the top part, the white line, and beyond 0.29 we get steady state. So this is a half bifurcation. But now when we have diffusion, then the system becomes much more complicated. So at first up to 0.12, which was our exponential decaying region, we have oscillations with spiral formation. The next region gives us random oscillations, and at this 2 point we have transients followed by stable oscillations, and beyond that window we have stable oscillations without spiral formation, and finally beyond 0.24 we have steady state. So this is the bifurcation diagram that actually tells us how changing the excitability of the system we can control the dynamics of the system. And that is why we can choose to call this epsilon as the excitability parameter. So moving on to the second part, the flux, this part. Can we change the flux in any way to control this? So we said that in the presence of translational diffusion coefficient, diffusion, we have simple this flux. But then, can we add something else? Can we put a thermal gradient? Will it bring any change to this kinetics of the reaction? Yes, indeed, it is going to act in this way mathematically, where gradient of T is the gradient of the temperature, and here the size of the particles is going to determine which way they are going to go. In the presence of an electric field, the flux can also be modified, and here the ionic charge of the ions, we are actually working with ions, positive ions, negative ions, so they are going to be influenced by an electric field or electric gradient. So let us theoretically see what happens to a reaction diffusion system under these different processes. So under thermal gradient, the heavier particles or the larger particles move towards the cold end and the smaller ones move towards the hot end. So using a simple reaction diffusion system, it can be found that the regions of absolute instability and convective instability can be brought in, can be attained by changing the thermodiffusion, delta T value. Under electric field, the same system of equations can give us an additional stationary state and another zone of absolute instability with negative velocity of the waves. Again, for the BZ, it is not possible with the current way we have. But if in our reactive system, by some means we have magnetically anisotropic systems, particles, which act under magnetic field, then it is possible to actually get pattern formation using magnetic field. This actually happens in the optical membranes that we have in our eye. Okay, optical things can be changed. Light can also change this kind of things. Okay, pattern formation is possible. And mathematically, we can show that if there is magnetic anisotropy in one of the variables, it is possible to get beautiful pattern formation. And finally, noise in the system, whether it be multiplicative or additive noise, can also bring about pattern formation. So all this was theoretical work on simple reaction diffusion system. Now, coming to our excitation waves, actual experiments, can we modify the flux? to control our excitation waves, to control their position. Let us see what happens in the laboratory. What if we use heat and electricity? We have different size of particles, and we also have ions in our busy system. So again, I have added both the experimental setup here. If we use hot water channels on two sides, it is possible to maintain a constant temperature gradient in the reaction. Or if we put copper electrodes on two sides and fix them to as anode and cathode, that too is going to give us a electrical gradient. So both can be used simultaneously, separately, perpendicular to each other, etc. So let us look at the simple case of a spiral. So first I will run the spiral without any gradient, which we have already seen. So we see that the spiral stays at a particular point, which is noted by the red dot. Now suppose I put a thermal gradient around our petri dish. So what is going to happen? You can very evidently see that the spiral is moving towards the hot end. It has moved away from its initial position of the red dot. Okay, so actually it moves in the direction of the field. If you go from cold to hot, it moves in the direction of the field under thermal gradient. So we can move excitation wave using thermal gradient. Using electric field 2, a two-dimensional spiral wave can be moved. It will again move in the direction of the field, or a three-dimensional scroll wave can be also moved. Okay, these were successfully done. Now we thought, can we change this field, the direction of the field, and move around our wave? Yes, indeed it was possible. If we take this center of the scroll, and mark it with this dot and start here. At first we had our electric field in this direction from right to left. Then the scroll was seen to move towards the left in a period of 21 minutes. In the next 63 minutes, we changed the direction of the field and we saw indeed that the scroll moved from the left and it moved to the right. It crossed its original position of 1 and went to 3. So this was a very interesting result. Then next we thought, why don't we move the electric field clockwise? So this was the direction of the electrodes at first, positive and negative. The spiral moved as predicted towards the positive end. Then we changed the electric field in panel B. So that was the next 35 minutes. So we took a window of 35 minutes for every. So we put 4 electrodes. First we ran the electric field along this direction, and then towards the right, then down, and then first towards the top, then left, then down, then right once again, A, B, C, D. And we did see that the scroll actually went around in a rectangular way, which was very interesting. So it shows that it is possible indeed to move an excitation wave under the presence of a gradient. So we said once while discussing about these waves that we know that these waves can get attached to a heterogeneity. What it means? So we have seen a scroll that slowly dies, right? It shrinks and dies. What happens if you put 2 glass beads on 2 sides? What is happening here is the scroll is just remaining like that. Of course, there is a initial change in the nature of the waves. It's become from completely circular to a little eye-shaped from here to here. If we take the time-space plot, you can see more clearly that it was this big. It has become a little smaller, but then it retains the shape. And theoretically, it is going to live on forever. So think in the scenario of the human heart, or any heart for that matter. If we have some obstacles, like the clot, one of the clot breaks the wave, we get a spiral or scroll, and then that wave actually arrests. The scroll is now living forever, right? It has got pinned. So that again is not a very good idea for 3-dimensional waves. So we thought, can we unpin them? Instead of giving a electric shock, can we unpin them by a milder method? So we tried giving a thermal gradient. So this is the same video we just saw a while ago in the past slide, where this was just where it was, right? It's not moving or changing anywhere. Now in the same system, after it has stabilized, we put a thermal gradient, hot and cold end. What we see is the end towards the cold side. The cold side is come off from the bead. So it has unpinned. That which was stuck is now unstuck. So this is for gradient 0, this panel. This is a cartoon. When we have the gradient on this direction, it is detached from the right side. When we have a perpendicular gradient, it detaches from both the things. I can show you the actual results. So you see, we are successfully able to unpin these waves by using thermal gradient. And once unpinned, because 3D waves are going to shrink and disappear. So that is what's going to happen to these scrolls. Okay? The scrolls are then naturally going to disappear. By electric field too, it was possible to unpin waves. And this over here on the right hand side is a mathematical model of the Luo-Rudy model of the heart. So they tried to use such a continuous electric field or electric gradient to unpin attached scroll waves to obstacles in the heart model. So what we are thinking here, or scientists are thinking here, is instead of giving an electric shock of a high voltage, which can cause scar tissues and damage the tissue, why not use something which is more mild and which may be able to unpin them, and finally it will reach the walls of the heart and we can maybe kill them. So finally, I would like to talk to you about, because I have been talking about that they are oscillating, they are rotating. So you might be able to think that are they anything like the rotors or the oscillators that we see in our physics systems, right? Like we see with the pendulum, like we see with our electronic systems. So if you want to look at these waves, or rather these rotors or singularities, as simple rotors or oscillators, and look at their interaction, what happens? In different kinds of systems, it's already known that vortices reconnect. So here is the reconnection of vortices. This is a work done in 1991 where two vortex filaments join and then they actually rejoin each other. So you see there were two on the top and bottom. Here we have one on the left and one on the right. Again, with vortex rings in air, they were able to show that two rings merge to form one large ring. So is that possible for our scroll waves? So indeed, we were able to do experiments. I'll show you the video where we saw the two scroll rings placed side by side actually attracted and they merged to form a large scroll wave. Now the lifetime of this merged wave is again many folds larger. In this case, it was 7 folds larger than that of the individual waves. So what was actually happening? If you think with the right hand thumb rule, that this is the direction of motion of the rotor, right? Then these rotors here, one is towards the down and one is pointing towards the up. So these two are going to actually annihilate, and the total flow of the rest of the filament is not changing. So this is the filaments, the two filaments which are different from two scroll rings initially, they join to form one large filament. We did several experiments with changing this interfilament distance and the size of the filaments or the diameter. And when we plotted them, we found that all these red dots are where they reconnected, where they joined, and all the blue dots are where they did not. So you can see that there is a direct, nice division, and this division occurs at 1.1, which is equal to the core size of a free spiral, or 1.09. So now if we try to remember that the scroll wave is actually made up of spirals. So when these spirals in this vicinity of the two scrolls, when they're close to each other, actually spirals which are counter rotating, when they come too close or within the core's size, then they annihilate. And that is what is happening. So that is what we thought was happening. Is it actually happening? In order to know that, we carried out several experiments, recent experiments with spiral waves. And with only 2 spirals, we interestingly found a zone of collision. This was much larger than, this was 1.09 millimeter. Here it is 1.95 millimeter. We found that they attract and annihilate. Beyond that, up to the size double of that, we found that they repel. And beyond that 0.39, which we called our critical distance, we find that there is a zone of no interaction. So the 2 spirals are where they exactly were in the beginning. So taking 4 spirals, also we see all these different similar kinds of behavior. So these are just 2 spirals. I have put the 4 there. So if this is the length or the distance between the center of the cores, depending on that distance, we can have, they are attracting. These are the tip trajectories. They attract and annihilate when they touch because they are counter rotating. If they are co-rotating, they will never annihilate. Okay, they will just move maybe as a pair or separately. When they are at a further distance, like here, then they move away from each other. And when they are in this range, then they just keep on moving around their circular course. Then we increase the number of spiral rotors, and what we found, that they could now join. I'll just show you. They could join like this. So 2 rotors placed like that, 2 spiral waves with 4 rotors. They could join like this. They could join between each other. So along L, they can join along D. They can all move away from each other, or there can be a symmetry breaking instability. So we plotted a phase diagram, just like our 2 spiral phase diagram. And what we found is, depending on the vertical and horizontal distance, they could annihilate in all these cases. There could be this blue dots, blue triangles are horizontal annihilation, means this way. The pink diamonds are vertical annihilation, like A. Here they are repelling, and when both the distances are beyond our critical distance, and this distance is exactly same as 16.4, beyond 16.4, that is the region of no interaction. So if you take any one, these yellow dots, orange dots are one numerical experiment, and you take one of them in the repulsive zone, and you see that they keep on moving till they reach this zone of no interaction. Okay, so these red cubes, red squares here are the ones which are in between the changing, the attraction and the repulsion, and there we get this symmetry breaking instability. So these are very interesting because our very symmetric initial condition does not expect us, or does not, we did not expect to find this kind of dynamics, but it might be happening because of some very local inhomogeneities, numerical in the case of numerics, and in the case of experiments too, we found that kind of symmetry breaking instability. So these are, so on the left, what I'm running now is a simulation, a video of a simulation where you can see what's the dynamics that's actually happening. The 2 scrolls are coming close, both in L and D, and finally there is a vertical annihilation. The top and the bottom annihilate. This is an experiment where we are seeing that 2 pairs of spirals, they annihilate. The top pair annihilates first and then the bottom pair. So this is kind of horizontal annihilation. So we tried to find the reason why the distance sometimes, when the horizontal is same or little more than the vertical, still they annihilate in the horizontal. And we found that the refractory zone, that is very important. So the zone, if we look at a spiral, this is the blue zone is the refractory of the spiral, and the top is the head. So they are moving away from each other. In that case, the 2 spirals would like to push away from each other in this direction. They still annihilate vertically, as seen here, when they are horizontally very far. But when we change the nature of movement, what if the spirals are not going that way but are turning like this? So you can see in this panel, the lower panel here, what if the two heads are pointing towards each other? In that case, comparative same value of L and D is going to make them actually annihilate along the head, so along the vertical direction. Okay, so now I have told a lot of things. I have told how we can control these waves. I have told whether they actually behave like rotors. But of course, there are many more things to look at, many more things that needs to be done. First is how general are these rotors in the perspective of nonlinear oscillators? Can they actually show us synchronization? I can tell you, yes, they do. We have got some results which show that they are able to show us chimera like structures, they are able to show us clustering. So that is really interesting for us. And then the challenge remains that in our busy system, till now, we have not been able to see the spiral breakup that happens just before cardiac arrest, the ventricular fibrillation. So that is something we want to achieve and we are trying hard to achieve in the experiments. Numerically, it is possible, but in the experiments, we would like to mimic the ventricular fibrillation, which we are still trying. And finally, after everything is said and done, after understanding the physics, the mathematics behind these waves, the main challenge remains that could we ever translate the scientific knowledge into technology? That is a much bigger challenge for the whole scientific community, and doctors should come forward and join hands with scientists in order to achieve that. And finally, will milder defibrillation techniques be possible once such technology becomes available? That will be really a life-changing scenario for many. And in this otherwise chaotic world, finally, this is something I always like to say, that there exists an underlying order. So, this order that I would talk about today is nothing that, even though chemistry seems to be far fetched from physics and mathematics and nonlinearity, it is indeed some similarity is there, and chemistry can act as a great tool in understanding natural phenomena and nonlinear systems. So I thank you all for your attention. It has been a long talk, and I'm sorry if I have ever lost your attention. I would really very much appreciate if I have questions. Thank you so much.
Thank you, madam. Thank you for your interesting talk. Now it is time for questions, clarifications.
Thank you very much, Sumana, for an exciting talk on the excitable systems. That's really very enjoyable. So I have some basic questions. So can one say, just like nonlinearity and dispersion, when they have fine balance, it leads to solitary waves, solitons. So similarly, when you have appropriate balance between nonlinearity and diffusion, you can have spiral waves and scroll waves. So can one make such a statement?
Uh, yes, there is a high chance that that is possible. Basically, without such competing terms, only one
Yes.
of them cannot give rise to this kind of patterns. So this
Yeah.
is possible, of course, as we have seen in just one single system, that every parameter space, it is not possible to attain it, but there is a possibility that if two things are competing, then we might be able to get something in between.
Yeah. Spiral waves, scroll waves. And considering scroll waves, so the underlying filament dynamics, so can you describe them in terms of Serret-Frenet equations? Just like vortex filaments hydrodynamics, you can describe in terms of Serret-Frenet equations of
Yes, it is actually
H-curve.
possible. I am not um, I'm very much a novice in this, but I do know, yes, it is possible to explain these vortices like it is done in fluid dynamics. It's essentially the same kind of thing.
Because in vortex filaments in hydrodynamics, when you make mapping on the space curves, then you can get solitonic equations
Yes.
in those cases.
Yeah,
So
they behave in the same way.
yeah, so I wonder what happens in the case of the scroll waves that you try to describe the filament dynamics in terms of
Yeah, actually, they
yeah.
would behave in the same way because the
Okay,
filaments essentially have the same physics.
okay,
The only
okay,
thing here in our particular system is uh, because of the positive filament tension, they tend to disappear. But again, in some biological systems, the filament tension is also negative, and there the chance of them expanding and breaking and all these things happens more. But of course, the physics is same.
okay, okay. Thank you very
Okay.
much. Thank you.
Thank you.
Sumona,
Yes.
yeah, I would love to ask questions after the students,
Yes.
um, but I'm not asking questions. I want to make few comments, basically. One of them is, it is well known that heterogeneity is not always destructive. It has constructive roles, like noise. But you showed examples that spatial heterogeneity can really induce uh, make a positive role in the sense of restoring the heartbeat. Yeah. So this is a really nice questions, and also you answered that the converting into technology is a long way, but
Yes.
that is the main target.
Yeah, that should be.
Yeah. So I like it. I like this pound. And second one, the when two scrolls are meeting, they collide and make a large scroll.
Yes.
It, it's my experience that it is similar to a phenomena to scroll to homoclinic orbit. Two homoclinic orbit like two scrolls. They also when they collide, they make a big circle. So the phenomena seems like this. I don't know. That is called basically it look like a gluing bifurcation.
Okay.
So gluing up to homoclinic orbits. It's like a,
Yes,
so
it's
this is
similar.
very common in double scroll system. So this one is also I like very much since I worked on it. And the third one is we did long back some work. Basically, Avadhes proposed, Avadhes that
Yes.
to counter rotating oscillators, if you couple them, they goes to death,
Yes.
amplitude death.
Yes.
So, so I think my student did an experiment in electronic circuit. So
Yes, I do remember. Actually, when I went through all your experiments, I listened to you in talks, all the kinds of electronics that is done, and especially with Professor Paramananda Punit's work on this camphor board. So that inspired me to work on synchronization of rotors. And we have beautiful results that show them clustering, forming chimeras. It's, I think I will be able to.
no, the thing is that the unfortunately that when I submitted first time in the physical review, Anthony Politi really said these are not counter-rotating oscillators. They are basically same oscillators. So this is all rubbish. All finally we proved it in electronic experiment. This is really true. You you have the same system, but they can rotate in the counter rotate as a
Yes.
work as a counter rotators, and you actually show us the same example, and
Yes.
they collide
Yes. They
to
collide and
go to
annihilate.
annihilate and also basically going to amplitude death,
And
right?
death.
Yeah.
Yes.
So thank you for nice demonstrations. I know
Thank
you are
you.
an excellent experimentalist in chemical oscillators, and we are really one of the two wonderful experimentalists in India, besides Puneet.
That's true.
So as an experimentalist, but my experiments is not so challenging because electronic experiments are much easier. But your experiment is more challenging. I understand.
It
So
is difficult. We are very
yeah,
slow for results, but we try.
yeah,
It's
yeah.
interesting. Thank you.
Thank you.
Thank you, Professor.
Thank you. Continue. Please continue with your experiments. Wonderful.
Thank you.
Okay, bye.
So Any other questions or clarifications?
I would really like some questions from students if you have anything.
Yeah, it will be wonderful if the student asks silly. You can ask silly questions. Any. Nothing is simple or silly. The question is question.
I think they are a bit hesitating.
Yes.
Okay, madam. Since I'm not getting questions, let us wind it up. So I conclude the session by thanking Dr. Usmana for giving a very wonderful talk on experiments on chemical oscillations. It is wonderful to see the chemical oscillations. Thank you, madam. Thank you for accepting our talk, accepting our invitation and giving a talk here. Thank you. Thank you, madam.
Thank
Bye,
you, sir.
Sumana.
Bye.
Nice
Bye,
to see you.
Thank you. Yeah, it was nice to see you too. Thank you, Professor Senthil.
Yes, thank you. I will write you the other details.
Sure.
Yeah.
Thank you so much. Thank you.